Hi, my name is Gloria Robert. Today we are discussing the topic biotechnology and genetic engineering. Biotechnology and genetic engineering. However, in this module, we shall be talking about biotechnology. Biotechnology. Objectives of lesson. By the end of the lesson, the learners should be able to 1. Define biotechnology. 2. State reasons microorganisms are used in biotechnology. 3. Enumerate the processes in the production of common bioproducts. And 4. Mention the important enzymes used in biotechnology. What is biotechnology? Biotechnology is the process by which biological organisms, systems or processes are applied in the manufacturing of industrial products. In another term, biotechnology involves the use of living things such as plants and animals to produce useful industrial products. Microorganisms are the main organisms that are used in the industry for the production of these useful industrial products. Commonly, we have bacteria and fungi. Now, why are these microorganisms used? That is because 1. They reproduce fast. 2. They produce certain complex molecules. 3. They have same genetic DNA code with other animals. 4. They have loop of DNA called plasmid. Plasmids very important in the production of certain insulin and protein. Number 5. They can be cultured or grown easily in the laboratory. 6. They are easy to store and occupy less space because of their microscopic sizes. And lastly, number 7. They have ethnical issues when used. Here is the structure of a bacterium. Please take note of where plasmids are located. Those are loop of DNA. Now the next thing we want to look at is the usefulness of biotechnology. Biotechnology over the time has been discovered to be useful in different areas of life. Number 1, in bread making. 2, production of biofuel. 3, production of enzymes. 4, fruit juice production. 5, washing powder production. 6, production of lactose-free dairy, such as milk. And number 7, production of drugs, and many more. So we shall be discussing these production processes one after the other. The first thing we are looking at is production of biofuel. Now, biofuel is a non-renewable and non-toxic fuel that is used in car and industrial machines. Now, unlike the normal crude oil products such as kerosene and petrol, biofuel is non-toxic and it is renewable. While petroleum or crude oil is, yes, we understand, it is not renewable and it is toxic. And that is why there is need for another source of fuel, and that is biofuel presently. Now, biofuel are ethanol or its mixture derived from the anaerobic respiration of plants, algae, and animal waste in the presence of yeast. So when these algae, plants, or animal waste are exposed to an anaerobic process, they can produce ethanol, which is the biofuel. Now the process include number 1, addition of amylase enzyme to corn, wheat, or sugarcane or any other source of carbohydrate in order to convert the starch in them to glucose. The next stage is to add yeast to the solution and then keep them in warm and anaerobic condition. Anaerobic condition means there is absence of oxygen, and in this process fermentation will take place. Now at the end of the fermentation process, ethanol is produced and collected to be used as biofuel. So the biofuel itself is ethanol. Take note. The next one we are looking at is bread making. Now flour is rich in starch and gluten protein, and flour is used in bread making. Okay. Now flour is very, very important because without flour you can't get bread. So there are steps involved in production of bread. Step 1, you have to mix the flour, the baking powder, and any other dry ingredients together. Add water in a bowl. Step 2 is to add yeast to prepare the dough. Don't forget that the yeast is the microorganism, is an example of a fungi. Step 3, you knead the dough properly and you mix them together. In this process, the amylase in the dough will convert the starch in the dough to maltose and glucose. Now that is the starting point for the process. Now step 4 is to allow the dough to rest for some time. Now during this process, By anaerobic respiration, the yeast that is added will produce bubbles of carbon dioxide that will get trapped in the dough, causing it to rise. So what makes the dough to rise is the carbon dioxide that is produced during the anaerobic respiration process. The next thing is that the gluten in the flour will make the bread to be stretchy and fluffy. Now, step 5 is to bake the bread in the oven. Now note that high temperature of the oven will kill the active yeast. So don't think that when you eat bread you are eating microorganisms such as yeast. No, the high temperature would have destroyed the yeast and also caused the ethanol that is produced during the anaerobic respiration process to evaporate. So here are images showing the steps used in the production of bread. The next one I want to look at is making of biological washing powder. Now, in the process of producing biological washing powder, certain enzymes are needed, mainly protease and lipase. Now, the enzymes are often obtained from thermophilic bacteria that are found in hot springs and have an optimum temperature they must work at. And the enzymes help to break down organic substances like oil, protein from food, especially on the plates that have been washed, or clothes, or any other substance that the washing powder are used for. And know that the enzyme used work at lower temperature. So very hot water used when the washing powder when you are washing the washing powder will cause the enzyme to denature. So the water must not be too hot. The enzyme must be used at low temperature. So let's discuss the next one, production of penicillin antibiotics. Now, penicillin is an example of an antibiotic, and it is made from fungus called Penicillium. So we have different species of Penicillium, and the fungus, which is the microorganism, is capable of killing many disease-causing bacteria. That is why they have to be produced in large quantity so that they can be used in making the different antibiotics. Now, this is done in an equipment called fermenter. Now, in this fermenter, the nutrients such as sugar, ammonia salt, and the penicillium itself are processed in the fermenter. Now, what is the function of the sugar? It is for respiration to take place. The ammonia is to provide protein for the penicillium in order for it to grow or increase in number. Then the feature of the fermenter itself is that it must have a pH and temperature monitor. So the pH must be right, the temperature must be right. Another one, it must have an inlet for oxygen to go in, and the oxygen is needed for respiration. And lastly, it must have a cooling mechanism so that the optimal temperature is not above, it's maintained. The next one we are looking at is the production of lactose-free milk. Okay, now we have milk that are rich in lactose. Now, lactase is an enzyme in the body that can digest lactose, sugar in milk, into glucose and galactose. So in our body, naturally, our system can digest lactose sugar. And what is used in digesting it is called lactase, is an enzyme. Now, some people, however, they are lactose intolerant. They cannot tolerate lactose in their body, especially the Asian descent. Okay, they find it difficult to digest dairy products such as milk, cheese, or butter. Now, how then can this lactose-free milk be produced? The process involves that the lactose milk is treated by passing it through beads of lactase. Lactase is the enzyme that can convert lactose to glucose and galactose. Now, the lactase can convert the lactose into glucose and galactose in this chamber. And then what happens is that the lactose-free milk is formed, and so it's collected from an outlet and then packaged and sold or used directly. Now, the next one I want to look at is fruit juice production. Now, fruit juice is extracted from fruits such as oranges, apples, mangoes, strawberries, mention them. So you can get fruit juice of different kind of flavor based on the fruit that is used. Now, to extract more fruit juice from this plant or fruit, an enzyme called pectinase can be added to the fruit. Now, what does the pectinase do? The pectinase enzyme breaks down a polysaccharide known as pectin in the cell wall of the plant. The function of the pectin is that it binds the cell wall together. So when it is broken down, it will free all the juice that are locked in the cell wall. So when the cell wall is broken down, more juice is gotten. So adding pectinase to fruit can also help to produce a clearer juice that is not cloudy, that is very clear and looks so nice to drink. All right, so that is the production of fruit juice. So take note of all of this process because they're actually needed when answering questions. Okay, all right, so here we've gotten to the end of the class. These are our students' activity. We have 10 questions for you to actually test your understanding or mastery of this topic. Please, if you don't understand, go back to the video, watch it again, listen to it, and I believe you'll be able to tackle all these questions and even more. Are you new to this channel? Please subscribe to my YouTube channel, click the notification bell, like my video, give me a thumbs up, and of course drop your comments. I'll be glad to respond to them. So at this time, you have really done well listening, using your time, and every other thing. So please promote my YouTube channel. Thank you so much. This is the time for us to mark. So we have Pyro to do the marking. So start marking. All right, our desire is to have 10 over 10, 100%. So I believe that is what you scored. If you don't score 10%, 100% or 10 over 10, go back to the video and listen to it. So at this time, I want to thank you for joining me in this class. Have a wonderful time.